Prokaryotic Cell Development in Caulobacter crescentus by Cody Evans, Margaret Peck, and Samantha Taylor. Just as humans and animals do, prokaryotes have to compete for limited resources in order to live. This photo illustrates the competitive nature of E. coli, the orange or pink bacteria slowly overcoming the green bacteria. Some organisms live on the move, hoping they'll run into new nutrients. Others retreat to extremes, where few other organisms can survive. Any way you look at it, organisms have had to evolve in different ways to give themselves an advantage.
Historically, bacteria have changed size, shape, and function to better adapt to their specific environments. Here we show the most common types: Micrococcus, Proteus, and Spirillum, to list a few. Well, that's cell development. Simply put, it's the change of form or function in order to better itself.
We'll take the example of Caulobacter crescentus. It is a single-cell, crescent-shaped, non-pathogenic bacteria found in both fresh and salt water and some soil environments. More importantly, it is found in extreme nutrient-poor conditions such as tap water. It's gram-negative and is one of the best models for complex cell development.
Why? Among many things, it has developed a dimorphic life cycle. During replication, Caulobacter crescentus elongates and asymmetrically divides into two different daughter cells, stalked and swarmer. Only the stalked cell can divide and form new cells. Yet, in order to ensure the longevity of the species, both forms should and must replicate. So, the swarmer cell morphs into the stalked cell, and then it is able to replicate as well. That's it. It starts all over again.
The swarmer cell has a flagellum and pili on one pole and is motile, whereas the stalk cell is completely and permanently stationary due to the holdfast extruded by its stalk. The swarmer cell swims for about 45 minutes before beginning the transition into the stalk cell. During this transition, it sheds its flagellum and attracts its pili. The cell then constructs a stalk at the site of the former flagellum. Now the cell is no longer motile. The stalk extrudes an extremely adherent glue known as the holdfast. Once it has adhered to a hard surface, there is no letting go. The holdfast has been proven to be 2 to 3 times stronger than the strongest commercial superglue. Essentially, it just sticks there and soaks up nutrients that are floating by. Why waste energy finding food when you can let it come to you?
CTRA is the master regulatory protein controlling the asymmetric cell division of Caulobacter crescentus. CTRA controls over 100 genes and binds directly to the origin of replication when it is phosphorylated, stopping chromosomal replication. The mechanism of phosphorylating CTRA is directly dependent on the histidine kinase CCKA.
CCKA works as a kinase, phosphorylating CTRA at the swarmer pole, and as a phosphatase, dephosphorylating CTRA at the stalked pole. When CTRA is phosphorylated, the cell is shaded blue. At the stalked pole, the protein DIVJ is present. It activates the CCKA DIVL complex so that it can dephosphorylate CTRA. However, in the absence of DIVJ, the two proteins PLEC and DIVL work together in allowing CCKA to transfer a phosphate group to CTRA. When CTRA is phosphorylated, it binds to the origin of replication, stopping initiation. The complete mechanism of CCKA regulation is unknown, and there is ongoing research to uncover more information.
From the localization of proteins in the asymmetric division to its uncanny ability to survive in nutrient-poor conditions, Caulobacter crescentus is one of the most powerful models for understanding prokaryotic cell development.